Is anyone going to tell me what they need me to talk about? Ruth? HW? Anybody? Tell me what I need to talk about so you guys can get the most out of this review session. I don't want to review stuff you already know. Okay, well, since no one's gonna, tell me what they need, I guess I'll just go through something and you guys can ask questions as we go along, I guess. So the first thing to talk about is, of course, we can talk about mitosis and meiosis. And so with those processes, there's a couple differences between them and there's a couple similarities between them. So first, let's talk through the cell cycle, and then we can kind of bring in the mitosis and the meiosis part of it. So with the cell cycle, there's 3 parts to the cell cycle. The first part we have is interphase. The second part is the M phase, and this is where we'll see the mitosis or the meiosis. And then the last thing we'll have is the cytokinesis. So it's important to understand that interphase and cytokinesis are occurring outside of the actual cell cycle. And so within interphase, there are 3 different phases. There's different parts to it. You've got G1, you have S, and you have G2. So G1's big important part is that this is when the cell is going to grow. So the cell grows during G1. The big thing that happens in S is that the DNA is replicated. And we can talk later about what happens in DNA replication if you guys are needing that. And then in G2, again, the cell grows. And technically the cell is going to grow throughout all of these phases. But there's only, the important part about S is that this is where the actual DNA replication occurs. And there's also some checkpoints. There's some places in which the cell cycle is going to stop. And so there's actually a checkpoint that's supposed to be a stop sign. There's a checkpoint that we have in the G1. And so the G1 checkpoint is meant to say, okay, should we divide? Do we need to divide? So should we divide? And like it's going to look for a kind of a growth factor or some type of signal to tell it to divide. And then at the end of G2, we're going to have the G2 checkpoint. Again, that's supposed to be a stop sign, but my handwriting's not the best, or my drawing's not the best. So we have the G2 checkpoint. And y'all can post questions whenever you have them. The G2 checkpoint verifies that all the DNA has been replicated and that the cell is an appropriate size to divide. So it's just making sure that we are ready to divide and that we should be dividing. And then, of course, we have the M phase. The big thing that happens with the M phase is that this is when our cell is going to divide. And I really shouldn't say the cell divides. This is where the nucleus divides. So this is when we're taking the DNA that we've just replicated and we've actually split it and kind of made it into 2. Maybe I should rotate over a little bit. That way you can see a little bit better. There's a lag, so it takes me a second to see if it actually is going to work or not. Yeah, it's the exact same. So the M phase, we'll talk about mitosis and meiosis in a second and kind of see what all happens in that. And then cytokinesis is our last thing that happens. And in cytokinesis, this is when the cytoplasm divides. So this is when we'll actually see the cell membrane pinch to divide the cell in two. Oh, I forgot a checkpoint. There's also a checkpoint in the middle of the M phase. This is the M checkpoint. And so with the M checkpoint, this is to ensure that all the kinetochores are attached to microtubules. We're good to go. Do you have any questions overall about the cell cycle? Okay, moving on then. So if we talk specifically about mitosis, there are three parts to it. You have prophase, metaphase, anaphase, and telophase. So the big thing about prophase is this is when the cell is preparing to divide. So this is when our sister chromatids will pair up, the chromatin condenses. So it's just kind of getting prepared and kind of getting ready for it to divide. Metaphase, the big important thing here, is that the sister chromatids align on the metaphase plate. So there's this region in the middle of the cell called the metaphase plate. And so what's happening is that these sister chromatids are lining up in the middle. And they're lining up so that, kind of if this was my sister chromatids, they're lining up so that one sister chromatid faces each pole. Because what's going to happen is that there are microtubules that attach to them. And those microtubules are going to pull these sister chromatids into opposite directions. And that's actually what's happening in anaphase, is that the sister chromatids are pulled apart to the opposite poles. And then in telophase, this is when I have my two new nuclei. So the nuclear envelope will form, and then the chromatin will decondense. And we mentioned before that there was an M checkpoint. That M checkpoint is in the middle right here. This is where that M checkpoint is. It's kind of to make sure that all of those microtubules are attached to all of those sister chromatids. Here's that M checkpoint. So, any questions on mitosis? So moving into meiosis then. So meiosis is actually going to take place in two different sites. So we're going to go through again prophase, metaphase, anaphase, and telophase, but we're going to go through it twice. So we still go through those same steps of prophase 1, metaphase 1, anaphase 1, and telophase 1, which would be the first round of division. And then we go through prophase 2, metaphase 2, anaphase 2, and then telophase 2. And so when we're going through this, kind of the important thing to realize is that the point of meiosis is to, in turn, reduce the chromosome number in half. So we'll end up making 4 cells in meiosis while we only made 2 cells in mitosis, because we'll go through that second round of division. So when I go through meiosis, I'm just going to kind of point out the things that are different from mitosis, because it's still the same idea where we're condensing, something's lining up on the metaphase plate, something's being pulled apart, and then we're making our 2 new nuclei. So the important part here is that in prophase 1, instead of our sister chromatids pairing up, now we've got our homologous pairs growing up. And so, in case you don't remember, homologous pairs would be your sister chromatids from the maternal side and the sister chromatids from the paternal side. So this is where our homologous pairs will kind of pair up together. This is also when an important thing called crossing over takes place. So crossing over is one of the things that's going to lead to genetic variation. And so this has to do with when the non-sister chromatids, so the sister chromatid from the maternal chromosome and the sister chromatid from the paternal chromosome, they overlap. And when they overlap, they exchange some of their genetic information. So metaphase, what's happening here, is that now we have our homologous chromosomes, or our homologous pairs, are what's going to line up. So in mitosis, we saw that it was the sister chromatids, but in metaphase 1 in meiosis, we're seeing that it's now the homologous pairs that are lining up. Again, in anaphase, this is our homologous pairs that are separating. So that's an important thing that you kind of understand, is that in meiosis 1, it's usually the homologous pairs that we're dealing with. Oh, I forgot to mention something. In metaphase, this is where we see that independent assortment. So that has to do with that the homologous pairs are independently aligning on that metaphase plate, and so they have this independent assortment that they happen to have. And then telophase, we have our 2 new nuclei. Now, important to understand is that at this point, the cell is already haploid. So it's haploid, and it does not need to go through a round of S phase because it's already haploid. It already has 2 sets of sister chromatids. It's already doubled up, if you may. And so thinking about meiosis 2, now this is when we're going to see our sister chromatids pair up. And meiosis 2 is very similar to mitosis. So sister chromatids pair up, sister chromatids are going to line on the metaphase plate. Anaphase, this is where our sister chromatids separate. And then now we have, again, 2 new nuclei. And again, the cells are haploid. So now I actually have 4 haploid cells. Because over here, I had sister chromatids, so they were already still duplicated. So, any questions on meiosis? We're okay on meiosis? Okay. And then cytokinesis is pretty easy. It just has to do with that the cell is pinching into two and kind of dividing. So where's my list of other things that we can talk about? Since no one's telling me what they need, so I'm going to kind of just roll and do whatever I feel like doing. I'm pretty much just going to go in order until I run out of time, which is 10 o'clock, because the It's Learning will only, the Instagram Live will only work for an hour. So thinking about genetics, kind of thinking about that there are lots of different kind of types of kind of dominance that we could talk about. So the first one is complete dominance. The second one is incomplete dominance. The third one is codominance. We can have epistasis. And then we can also talk about multiple alleles. So with complete dominance, this has to do with that your heterozygous and your homozygous dominant are going to look the exact same. So if I have AA and I have A little a, these are going to have the same phenotype. Because the dominant allele is able to completely be dominant and completely mask the recessive allele. So now it's important to understand that recessiveness doesn't mean that it's just not there. It just means that it may make a non-functional protein, or it may just mean that the dominant protein is going to kind of overlay it or kind of cover it up. So with incomplete dominance, you're going to see that there'll be three different phenotypes. So AA, A little a, and then little a are going to give you three different phenotypes. And so what we find is that the heterozygous tends to be kind of an intermediate or a blend between the two. So the good example that we have with this has to do with the snapdragons. So like if AA is red and little a is white, then what we'll find here with our intermediate is that it's going to be pink, because it's an intermediate between the red and the white. So we kind of see a blending of the two. Codominance, again, you're going to see all three of them. So AA, A little a, and little a is going to give you three different phenotypes. So with this, we could talk about, you could have red, you could have white, but the intermediate is going to be red and white. So we kind of talk about this with roan cows. There's these cows that have like red and white spots, and it has to do with that they're the heterozygotes. Epistasis is kind of weird. With epistasis, you have two different genes, and then one controls the other. So what we're going to see here is that the first gene, let's say that it's pigmentation. So if you have capital P, or capital P, then you'll pigment. And then if you have little p, then you'll have no pigment. And then it's going to look at the second allele. So let's say that we're talking about AA and A little a, that would make it red, and little a makes it white. And so what would happen is if you have, we shouldn't do white. Let's make it pink instead. So if you have the dominant allele for pigment, then you'll see the red or the pink. But if you have the recessive for the pigment, your homozygous recessive, then you would end up being a white flower because you wouldn't have that pigmentation. And then multiple alleles is kind of cool because it has more than two alleles. So we usually look at this at blood type. So I'm going to move away from my A's for a second here. And so what we'll have is that you could have IA as an allele, you could have IB as an allele, or you could have a little I. So as you notice, there's 3 different alleles here versus in the previous examples where we had a capital A or a little a. Now we're looking at 3 different alleles. And so what ends up happening here is this can give you more phenotypes. So if you have capital A, B, then you're going to have AB blood. If you have the IA and the little I, then you'll have A blood. IB little I, you'll have B blood. And if you have 2 little I's, then you'll have the O blood. So it kind of shows you that there can be lots of different things happening. And something I also didn't mention, but could show up on your pedigrees, is that if you happen to see a mother that's affected and every single one of her children is affected, it could be mitochondrial, in which the error happens to be in the mitochondria, in the DNA of the mitochondria. And so since the mother donates all of the mitochondria to the offspring, then you're going to find that all of the offspring will have that mutation. So do I need to do practice problems with these genetics things, or you guys feel okay with just the different types of genetics? And again, there's a lag, so it's taking me a second to hear you. Could you do one, please? Hector, is there a certain one you want me to do, or is it just whichever one I want to do? I guess Hector doesn't really... Oh, wait. Whichever you feel people struggle with the most. I guess we can blend some together. We could talk about incomplete dominance and talk about epistasis, which will show how to do all the math problems. So we'll do a combination of incomplete dominance with epistasis. So if we have a, let's see, we have a flower that follows incomplete dominance and epistasis. So the cross is going to be between a red flower and a white flower. If they get the 2 little pigments, then they're going to actually be yellow. Okay. And so since I see here that I have a red flower and a white flower, and I know that I'm doing incomplete dominance, I know that this one has to be homozygous red, and this one has to be homozygous white. And then because of the fact that I've got the epistasis, I know that it's got to be, we're going to assume heterozygous just so that I can do this cross. So we'll do 2 different Punnett squares because we're looking at 2 different genes. Anytime that you have a dihybrid cross, or you have some type of cross that involves more than 1 gene, you always want to just do simple Punnett squares. You don't want to try to make your life complicated by doing one of those big Punnett squares. So we'll first do our red, cross with our white, and you can fill these all in, but this is going to be all pink, which probably shouldn't have been the best example for me to use. And then here we have our heterozygous. Okay, and so what we're going to see is that I need to change the question. How about we make this a pink one with a white one? So I make that CW. And then we'll make this the CW. Okay, so that makes that CW, makes that CW. Okay, so then that makes this 1/2 pink, which is CR-CW, and then 1/2 white, which makes this CW-CW. And then here we're going to see 3/4 will be pigmented, so we'll see the color that's supposed to be. And then 1/4 will be no pigment, which we said at the beginning is going to be yellow. And so if we wanted to do the math for this, which I see that I'm not on the screen anymore, we're going to multiply this together. And so if I want to know the ones that are pink, I'll say, okay, 1/2, because there's 1/2 of them that were pink, and then 3/4 that were pigmented is going to give me 3/8 of them that are going to be pink. If I want to know how many of them are going to be white, I'll say, okay, well, there's 1/2 times 3/4, which again gives me that 3/8. And then this no pigmented one, we said is going to be yellow. So all of these, it doesn't actually matter whether it's pink or white, all of them will end up having no pigment if we have the recessive. So we can say 1 times 1/4, which will give me 2/8. And I just went ahead and got the same denominator, so it would be 1/4, and if you multiply that by 2, you would get 2/8. So this ends up giving us a 3:3:2 ratio, or a 6:6:4 ratio, which is a different ratio than you guys are used to. But that is how we would, I guess, work it out. Any questions about that, Hector? Since you're the one who asked for me to do a question. We're okay? Takes a little lag, so sometimes I don't know how much lag there really is. You're okay, thank you. All right, so moving away from genetics, we can talk about replication, I guess. So since no one actually submitted any questions, so I don't really know what y'all are needing. So with DNA replication, it's going to go through a semi-conservative model. So what that means is that genetic linkage or fruit fly. You want sex linked? We can talk about sex linked problem in a second. Let me go through the DNA replication, and then I'll go back and do a sex linked problem. So with DNA replication, we said it's semi-conservative. What that means is that you're going to take your parent strand and you're going to separate the parent strand so that we can conserve the parent strand and then also have a replicate with it. And so if this was my DNA, there is a 3' end and a 5' end, and then of course the 5' and the 3'. And in case you need a reminder of it, the 5' is where our phosphate is, and then the 3' is where the hydroxyl is. And the hydroxyl is of course on that pentose sugar that you have for your DNA or your RNA, whichever one it is that we're working with. Right now we're working with DNA, so of course that would be the deoxyribose for that sugar. And so we're going to have a couple different things at play here. First thing is you're going to have this enzyme called helicase. So helicase is an enzyme that is going to break the hydrogen bonds between the nucleotides. And so what this means is that originally the two strands were attached to one another. There was hydrogen bonds in between them. And so we break those bonds, so now we're able to separate it. So as you see, I went ahead and separated my two strands. So that was because helicase went and opened it up, okay? This location right here is called your replication fork. And at the replication fork is where the helicase is working to actually open it up. So the second thing that we have to talk about is primase, and I will come back to you in a second, Hector. We have primase, okay? Primase is going to act to add an RNA primer. And so by adding the RNA primer, this is because DNA polymerase cannot build off of nothing. So we need to have a primer added in order for us to be able to make the strand. So our RNA primer, let's make that, I don't know, purple. Okay, so we're going to have our RNA primer. Okay, again, it reads it 3 to 5, so it's going to make it 5 to 3. And then here we'll have our second strand. Okay, so that purple would be my primer. And again, we've got our 5' and our 3'. And this is my primer, 5', 3'. And I'm going to draw an arrow just so that we don't have any issues with this, okay? So Hector mentioned that he thought that there was another enzyme before that unwinds the DNA. There's not really another one in front of the helicase. These are kind of all working at the same time. So the helicase is opening, single-stranded binding protein is binding to it to keep them from coming back together. There's a lot of things that are happening all at once. So what Hector is mentioning is something called topoisomerase. And topoisomerase is going to be upstream from that replication fork. And its job is to relieve the strain. So those of you who are not my students, what I do in class is I take someone's shoe and I twist the shoe up so it makes a double helix, and then I hold the shoe and I pull the shoestrings apart. And so you'll notice that when you pull those shoestrings apart, that twist becomes like super coiled and it causes a strain. So the job of topoisomerase is to kind of relieve that strain so nothing's going to happen to my DNA and my DNA doesn't become super coiled. So that's what I think Hector was talking about. Hector, correct me if I'm wrong about what you were thinking. So again, after primase, we're going to have DNA polymerase. And so the job of DNA polymerase is to actually add the DNA nucleotides to make your strand. And so let's make it, I don't know, orange. So after our primer, we're now able to build off that primer and to build our DNA strand. So now you'll notice that I've drawn these in two different directions because here's my DNA, here's my DNA. You'll notice that one of the strands is going into the replication fork while the other strand is moving away from the replication fork. So they have funky names for these. The strand that moves directly into the replication fork is going to be called your leading strand. So the leading strand is the strand that is continuous, okay? So right now at this moment, our replication fork is only open to this much. But if we keep allowing helicase to move, it's going to keep opening up this strand, and it's going to keep opening it up downward. And so this DNA is able to actually keep moving as that replication as the replication fork keeps opening. It's going to keep moving towards it. So it's a leading strand. It's continuous. It never has to stop because of the fact that it's run out of nucleotides. Versus we're seeing over here that we have our lagging strand. So our lagging strand is going to be made discontinuously. And so we actually have a term because you're going to notice that there's this little fragment here. And if we opened up again, then I would have another fragment that's right here. And so the leading strand, as we said before, is going to be continuous, while the lagging is discontinuous. And so the lagging makes these strands, these little fragments, which are called Okazaki fragments. And so after this whole process, after we've gone and made our strand, you're also going to use another enzyme that's called ligase. And ligase's job is to seal the phosphodiester linkages. Between your nucleotides. So once I have another strand here, I would have to put DNA ligase in there to allow these two strands to now be attached together. And so with prokaryotes, they wouldn't really need to use that ligase because, well, I guess they would. They would use it once because they start at one origin and they move the whole way around, and then they've replicated their whole strand and they're good. Versus with eukaryotes, we've got multiple origins of replication. So when we run into another strand, DNA ligase would then, of course, bind and do that. Okay, so do we have any questions about replication? How are PCR machines involved with DNA replication? PCR is a technique that's used to be able to make multiple copies of a certain strand or a certain fragment. So with our PCR, there's 3 steps to it. The first step in PCR is going to be where it heats up. And so by heating it, what it does is it takes that strand and pulls it apart. And then the second step that you're going to have is going to be the cooling step. At that cooling step, the primers that we already have in the machine is going to bind to our DNA. And then you're going to go through the polymerase step, which is where we're going to use the Taq polymerase. And we'll end up making our strand copy. And so what ends up happening from this is that I can go through this process multiple times and I can keep making multiple copies of the segment. So the first time I start out with 1 strand and now I have 2, and then the 2 becomes 4, and the 4 becomes 8, and the 8 becomes 16, and 16 becomes 32, and 32 becomes 64. And after a long period of time, now I've got tons of copies of this fragment. So like, you know, if someone was murdered and they scratched the culprit, some of that DNA would be underneath their fingernails. Well, you would want to be able to test that DNA. And so what they can do is they can do a PCR to amplify and make more copies of that DNA, so they could then test it and be able to determine the identity of the person. Any other questions about PCR/DNA replication? Did I answer your question, Hector? Okay. So somebody asked a little while ago for me to do a gene linkage problem. I guess we could talk about a sex-linked trait. And so real quick, just kind of thinking about linkage or just kind of linked genes. This means that they're going to be on the same chromosome. So we can kind of approach this 2 different ways. So we could do a sex-linked problem. So sex-linked just means that it's on the X chromosome. And so if we were to talk about, I don't know, how about hemophilia? So you have a woman with a hemophiliac father who has a normal male that she mates with. And so what that means is that when we do our cross, males are on the top. So since he's normal, he would just be X, Y. Since she is normal, but she had a hemophiliac father, we know that she's going to have the recessive allele because her father would have passed on that recessive allele to her. So when we do that cross, you're going to find that we'll end up with 1 normal female. We'll have 1 normal male. We'll have 1 carrier female, like the way the mom was, and we'll have 1 affected male. Another thing that you could see with our sex, like with our linkage, is if 2 traits are linked together, as I said before, they're on the same chromosome. And so if you did a cross, I don't know, let's say we have yellow, wrinkled, and green smooth. And so if you cross those 2 together and you end up with 400 that are yellow wrinkled, 400 that are green smooth, 100 that are yellow smooth, and 100 that are yellow wrinkled, because of the fact that most of them look like the parentals, that would tell you that those traits are linked. And then these 2 would be considered our recombinants. because of the fact that they are a combination of the parent. We've got the yellow with the smooth, the wrinkled with the green. I think I wrote something wrong. Sorry, this should be green. Sorry, green wrinkled. Sorry. So we've got our green and our wrinkled together and our yellow and our smooth together. Those would be combinations of the parents. And so if we wanted to do what the recombination frequency is, we would take the number that are recombinant, so 100 plus 100, over the total number, which is 1,000. So we would get 200 over 1,000. So that would tell me 20%. So that tells me that these are 20 map units apart. Does that answer your linkage question that you had? I actually don't know who you are. But I'm going to call you Sal. Okay, I got a heart, so I'm assuming that means we're good. Okay, so after replication, we could talk about transcription and translation. Transcription is very similar to replication. I'm a mentee in China. Hi, mentee in China. Why are you following me if you're a teacher? So with transcription and translation, again, this is very similar to the replication, except for the two strands aren't going to come apart. They're just going to pop back together after they've made the copy. So you're just going to use RNA polymerase and it base pairs like normal. So I don't really think that you guys need a diagram of your replication, of the transcription, because it's a very simple process. But you may want one of kind of translation and kind of how that translation takes place. And so the mRNA is going to come out of the nucleus and is then going to go to a ribosome. Ribosomes all start out in the cytosol. And then once they get a signal to go to the membrane, then they would go to the membrane. But so within our ribosome, we've got 3 sites. You've got your A site, your P site, and your E site. And of course, this is our small subunit, which is going to bind to the mRNA. And then this is going to be my large subunit, which is going to bind to the tRNA. Hi, Morgan. So at the A site, this is where we're going to, I tell my students, we're going to add the next amino acid. So a tRNA is going to come in. So here is my tRNA. It kind of looks like a clover leaf. And what it has on it is it's got the amino acid. And so the tRNA is going to move into the A site, and at the A site is going to base pair up with the codon. So if I have, AUG here, then we're going to have UAC here, because these ones are going to base pair, which will cause this tRNA to come and bind to the A site, okay? So then what happens is the whole thing shifts over, okay? And so it's kind of hard to show a in motion thing, but we're going to try this. So that tRNA that we just had a second ago is now over here at this P site. And so at the P site, I always tell my students that this is where we're going to have the polypeptide. So this is where we're going to hold that growing strand. So the polypeptide has moved over, okay? And now I'm going to add a new one. So as we said before, this was AUG, this is UAC, and I drew it on the wrong thing, but you get the idea. Let's do a, I don't know, UCC. And I'm not looking at a codon chart, so if I'm wrong, just go with it, because I don't actually know all the amino acid stuff out my head. But so we're going to have another tRNA is going to come in. And let's use pink. So when it comes in, okay, it's going to come in base pairing in. So this kind of moves up into this site. So we have that AGG that it has. And what's going to happen is that the bond right here is going to break. It's going to form a new bond right here. And so now when the whole thing shifts over, now we're going to have this empty tRNA that doesn't have an amino acid on it. Here we've got the one that we just made a second ago that's got the blue strand and then the pink strand. And then a new one's going to come in to the site. So it's going to kind of keep jumping in. And so the thing I tell my students about the E site is this is where it's going to exit. And so we bring in the new amino acid at the A site. That new amino acid is then going to pair up with a growing polypeptide. So then when it shifts over, now we've got the polypeptide being held at the P site. And then the E site is where the empty tRNA is going to exit. Does that make sense? And this is like a dynamic process. It kind of keeps shifting down and keeps moving until it reaches a stop codon. And the stop codons, you won't have to know them, but they're relatively easy to remember. UGA, UAA, and UAG. And then the start codon is always AUG. Now Hector says what are the two types of ribosomes? You could have a bound ribosome or you could have a free ribosome. Bound ribosomes mean that they're bound to a membrane, so that would be like being bound to the nuclear envelope or being bound to the ER. And so once the ribosome gets the signal to bind to the membrane, that's when the free ribosome will become attached and become a bound ribosome. And then the free ribosomes just kind of chill out in the cytosol, and they will basically make cytosolic proteins. So the big difference between them is that the bound ribosomes make proteins that are inside of membrane, and the cytosolic ribosomes make proteins for the cytosol. And I believe that it's actually part of the review today. Like I talk about the difference between bound and free ribosomes. So look at the AP review for today. I believe I have an explanation there on those. Okay, we've got 10 minutes left. I guess we could talk about DNA technology unless y'all have more questions about translation. So with DNA technology, there's a couple different kind of examples that we kind of use. One we can talk about is gel electrophoresis. Another we have is the transformations. And then another was PCR. And this thing was going to end at an hour, so I have 9 minutes to get through this. If I'm not done with it, then it's just going to cut me off, so I'm sorry. So we're going to try to jump through the gel electrophoresis and all this stuff real fast. So gel electrophoresis, what it is that it is a porous gel. And then you're going to cut the DNA using something called a restriction enzyme. So the restriction enzyme is going to look for certain sites and it will cut at those sites. So what will happen is that if I've got similar DNA, then it's going to cut at similar sites. So I'll be able to observe the banding patterns to see where the similar DNA is. And so when we do our gel, we're going to have these wells at one side. This is where we're actually going to put the DNA in. So this is going to be your negative side, and then this will be your positive side. We'll set up electricity, and electricity is going to actually pull the DNA. The DNA is negatively charged, so it's going to get pulled toward that positive end. So you'll find that we have our larger fragments here and our smaller fragments there. And so as I was talking about with the banding pattern, if I have, I don't know, these bands here, and then I have this, and then I have this. This can tell me that what I have is a individual that is heterozygous, while this one could be your homozygous recessive, and then this one could be your homozygous dominant, because of the fact that you see the same banding pattern between this one, and then you see the same banding pattern between this one and this one. So you know that there's a similarity in those strands of DNA. And so that's an example that they used on a previous AP exam, was they used the gel and then you were figuring out the banding pattern of it. With a transformation, you're going to transform bacteria. And so you're going to get the bacteria to take up external DNA. And so traditionally what you'll do is you're going to select for certain DNA in this process. So we'll have a couple different plates. So we'll have just the LB plus, LB minus, LB amp plus, and then LB amp minus. And so you would expect a full lawn of the LB plus and LB minus. The reason why you would expect a full lawn is because of the fact that there's nothing to restrict their growth. There's no ampicillin to cause them to die. So you would expect full growth. If there's not full growth in these two, that's going to mean that you made an error in the experiment. In the LB amp plus, you're going to find colonies. This shows you that these are the individuals that were able to be transformed, and thus these are the only ones that can grow in the presence of the ampicillin. This shows that we selected for the AMP-resistant bacteria. And then your AMP minus, you should find that one to be an empty blank one, because of the fact that these individuals were not given the plasmid to allow them to be resistant, and there's ampicillin in the environment, which should have killed all the individuals. And then PCR, we just talked about a moment ago, which stands for polymerase chain reaction. And this has to do with that we're going to go through repeated steps of heat, cool, polymerase to make multiple copies of a DNA fragment. Whew, 5 minutes to spare. What other questions do you guys have? Are we good? Any other questions that you have? Yes, I'm good. Thank you. Oh, you're welcome, darling. I think I'm going to take pictures of these papers, and I'll post them on my Instagram, because I think that maybe some of it's hard to read with the way that the aerial shot is, but I don't really know any other better way to make the aerial shot. They're not required to know any of the diseases. If they have a disease, you're either going to have to look at a pedigree to determine the inheritance of it. So if you see that it skips generations, you're going to know that it's some type of recessive. If you have a father that's affected, if you have a mother that's affected and the sons are affected, you could probably think that that's going to be sex linked. You kind of have to follow the pedigree to figure that out. But they should give you background information or some diagram that would help you to explain it. I truthfully don't know what's going to happen with it being all free response. I'm going to try to find some example problems that I can use to do some live sessions to be able to prepare my students appropriately with how to determine that information. But with them all being online, it's gonna be not knowledge based. It's going to be an exam in which you're getting information, applying information. So it's not going to be like any of the traditional questions that we've ever seen. And I think I have about a minute left. So I hope you guys have a good day. And I'm going to try to save this so that it won't go away and you at least can access it if you didn't get into the chat. Bye, guys.